This podcast is brought to you by Bioceuticals Clinical Services. For more information, go to bioceuticals.com.au and click on the Education tab. This is FX Medicine. I'm Andrew Whitfield-Cook. Joining us on the line again today is Annalies Corse. She's a medical scientist, lecturer, naturopath and author who's based in Sydney. Annalies has worked as a medical scientist at Charles Sturt University, the Australian Institute of Sport, Australian Biologics and both public and private hospital pathology laboratories. Annalies has been a practising naturopath and lecturer since 2008. Annalies is a past board member of the NHAA and is on the scientific advisory board for the Mind Foundation. Most recently, Annalies has contributed to Leah Heckman's textbook called Clinical Naturopathic Medicine, due for publication in 2018. Welcome back to FX Medicine, Annalies. How are you?
I'm really well, Andrew. Thank you for having me again.
My absolute pleasure. And today we're going to be diving into COMT and CBS genes. So I will ask you to lead me in this because I am not an expert. Firstly, what are we talking about here with COMT and CBS genes?
So most of the practitioners and students listening may have heard of them as being related to genes in our, not necessarily the methylation pathway, but biochemical pathways associated with methylation. Now, the problem is that these days, with all the sequencing tools that have been developing after the Human Genome Project, because it's now faster and cheaper to test all of these things going on in our DNA, it's blurred the lines with some of our terminology. So the definitions of mutations around COMT and CBS or polymorphisms associated with COMT and CBS is becoming problematic, not just for scientists, because there's a huge debate in the medical literature about what term to use, but also for clinicians in practice. We need to know, well, is this something I need to test for? Is this something I need to treat? These two particular enzymes or pathways, when I was studying naturopathy, we didn't cover them. There was simply not a single lecture even on this, even in our advanced nutritional biochemistry subjects. So COMT is a gene for an enzyme known as catechol-O-methyltransferase, and it's particularly involved in the inactivation of the catecholamines and some neurotransmitters, so things like adrenaline, noradrenaline and dopamine. It's also involved in the degradation of the catechol-estrogens. So when we're degrading our estrogens in both the male and the female body, they need to be degraded in the liver, and COMT is involved in that as well. It's also involved in the degradation of bioflavonoids as well. So it's as an enzyme or as a gene coding for a protein product, in this case an enzyme, it's part of that group of methyltransferase enzymes, because what it's doing is it's transferring methyl groups, and this is why as an enzyme it's linked with methylation pathways, because it's taking methyl groups from that methionine cycle, particularly from SAMe, S-adenosine methionine, that people would remember from that methionine cycle, and it's taking those methyl groups in order to fulfill its role as a methyl donor and a degrader of catecholamines, catechol-estrogens. The second one that we're going to be talking about today is cystathionine beta-synthase. Again, there's a gene for this, the CBS gene, that codes for the CBS enzyme. Now, this particular enzyme, again, is involved in some of these methylation pathways or biochemical pathways that are sort of orbiting around that methylation methionine cycle that everyone knows as being the most famous one. So CBS is actually the first enzyme, or the step 1 enzyme in the transsulfuration pathway. So you might remember that one
right.
as the homocysteine down to cystathionine.
Yep.
And this is a gateway. This is an alternative pathway to methionine resynthesis, and it's the only route for homocysteine elimination. So the downstream products of this pathway include our sulfates, taurine, glutathione, so endogenous antioxidant synthesis, and ammonia. So I guess a lot of listeners will have done a lot of maybe postgraduate study around, NTHFR and those types of SNPs. These ones are starting to be mentioned, but they're still quite new. So in a nutshell, that's what they are.
Can I just ask, with regards to COMT, we always talk about it with regards to, estrogen metabolism and other neurotransmitter metabolism. What's the active site? Is it in the liver only, or is it elsewhere in the body as well?
No, there's two. There's actually there's two types of COMT that are synthesized. There's one which is the soluble form, and then there's one that's known as membrane-bound COMT. Now, the soluble form has more of an activity on, or it predominates in the liver, the kidneys, and the GIT. So this is the one that's more associated with things like methoxyestrogens and catechol-estrogens and the degradation of estrogens.
Yeah.
The membrane-bound COMT is the major species in the brain. So they're slightly different in terms of their amino acid structure, and they have different sites and therefore different receptors in different parts of the body. But they would be, if we would actually look at the amino acid sequence, they'd be extremely similar with only subtle changes there to distinguish between the two types. Okay,
but when you're talking about a SNP, wouldn't you then be looking at a SNP that's relevant for either soluble or membrane-bound?
Yes, so there's different ones. There are many, many SNPs associated with COMT. It's not just one. There are some that are more common and some that are studied more. And those SNPs usually involve a difference or a substitution of valine to methionine. And usually you'll see that occurring in both the soluble and the membrane-bound.
Ah, okay.
So it can occur in both.
Yeah.
That substitution might actually occur in different amounts in the S-COMT, so the soluble COMT, and at slightly different numbers of substitutions in the membrane-bound COMT. Or position. It might even be position. So the valine might be, the methionine might be replacing the valine at a specific position along that peptide chain, and it differs between the S-COMT and the membrane-bound COMT, if that makes sense.
And what about the CBS gene?
Okay, so with the CBS gene, again, there are many SNPs that have been, discovered or associated with CBS. Again, this is one where we don't really understand just how clinically significant the SNP is. The most important thing with this one is that it is, again, it's a very, very subtle change where you have a substitution of one amino acid to another. The interesting thing with CBS and where it differs a little bit to COMT is that the SNPs seem to either upregulate or downregulate the CBS activity. And some SNPs are associated with an upregulation and some are associated with downregulation. And that can also be manipulated by what's going on elsewhere in those methylation cycles. So I guess if the question is how significant are the changes within an SNP, the changes are very, very subtle.
Does that lead to the sort of questionable significance of the CBS gene in a clinical situation?
Yeah, absolutely. Because I guess the problem with all of this is it's all very, very new. Even though these SNPs have been known about for a few decades now, we're still in the infancy with the research with regard to what does this actually mean in a clinical situation. A lot of SNPs, not just in COMT and CBS, but elsewhere in the wider genome, they might not have any clinical significance whatsoever. It may just mean that you metabolize a particular drug differently, or you metabolize a particular hormone differently, or you might have an increased resilience to a particular set of pathologies. So SNPs in general are not always associated with doom and gloom in terms of pathophysiology. And I guess the problem we now have is that SNPs are sort of almost being associated with problems. That's not always the case. They are subtle changes. You would never just want to associate SNPs with disease. It may just mean variance. And that's the thing with the genome. All humans are different. And, you know, when you look at the Human Genome Project and its significance and its findings, you know, greater than 95% of human DNA between different individuals was the same. The difference is in that 5 or less %. And that's often where a lot of these SNPs are. And that's what makes us different. It doesn't necessarily make us these genetic mutants that are going around having an increased susceptibility to terrible diseases. That's not always the case.
What are the conditions for which these SNPs are clinically relevant then?
Okay. Well, there's really broad categories. And first up off the bat, there's probably a bit more evidence for conditions associated with COMT than there is for CBS in general.
Right.
So CBS, if we discuss that one first, that one seems to be more associated with fertility risks. There's a group of genes, well, CBS is one of the genes involved in folate metabolism. So I tend to think of it as this less famous cousin of the MTHFR gene and the SNP associated with that.
Right.
So there are fertility risks in terms of increased risk of miscarriage, recurrent miscarriage. When a pregnancy is established, there's often an increased risk of pre-eclampsia. And generally folate metabolism in general, so the ability to activate folate for the growth and embryology of the growing baby, that does depend on this CBS gene. So CBS and fertility, there seems to be a fairly well-established link there. Again, someone would probably need to be homozygous for that SNP, so they would have had to inherit 2 copies of the SNP, one from their mother and one from their father. That is going to result in an increased likelihood that that SNP is going to be problematic, as opposed to someone who's got a heterozygous status for the CBS SNP. Usually studies try to quantify how much of a increased or decreased enzyme activity there is based on heterozygosity versus homozygosity.
Yeah.
It is a tricky thing to do. It's not established very well in the literature yet.
I guess with things like certainly fertility, you tend to hear of people having recurrent issues with fertility before they actually delve into the reasons why there's issues for fertility. But pre-eclampsia, I mean, that can be a really devastating condition. We're talking about safety of the mother here and indeed the baby.
Yes.
Where do you think the relevance is for pre-testing and or, you know, testing if there's an issue, you know, testing for subsequent pregnancies, for instance?
Well, I think first and foremost, when someone is pregnant and they're in those early days, usually with her GP or her obstetrician, there's a lot of questions around family history. So they're asking about diabetes, did your mother have pre-eclampsia, clots, all of the things that can be associated with negative outcomes in pregnancy. So usually things are questioned in those early stages of the pregnancy, but that doesn't really seem to address the problem, because from our point of view in naturopathic medicine, there's something that we could be doing beforehand in preconception care. So to my mind, we would need to be at least thinking about this in the preconception phase. And we all know that preconception is not a part of primary medical care.
No.
It's something from naturopathic medicine. So it's up to naturopaths, I think, and nutritionists and herbalists or whoever it is that's doing the preconception work to identify with the lady who's thinking of conceiving, well, what did your mother have pre-eclampsia? Do you have any sisters? Did they have pre-eclampsia? What about your grandmother? And if you're getting strong indications of a family history, then it might be worth thinking about things like MTHFR, CBS, SNPs. Whether or not you test, that might be something that you decide to do or you don't decide to do. But it's definitely a question. And I think for us, because we're working in the world of preconception, that's when it should be coming up, because they're not really going to be getting these kinds of questions from GPs or obstetricians.
Look, I've got to ask the question here, though. Like, I'm always concerned about over-testing, but I'm also concerned about under-testing where it's relevant and where you can pick up a potentially serious condition and do something about it very early rather than waiting till the horse has bolted. So when we're talking about something like CBS gene, and you spoke about risk factors in the mother and siblings, what if they're heterozygous though, and this patient is homozygous only by way of being the, 9-3 to 3-1, the unlucky person that gets both chromosomes?
Yeah. So they've got the homozygous for the SNP.
Yeah.
Yeah. Look, I think if there's any family history, and it's a strong family history, say the woman's mother, and if she had strong clinical conditions in her own pregnancy, such as hospitalisation from pre-eclampsia, diagnosed pre-eclampsia, embolus formation during her pregnancy, they are serious conditions.
Yeah.
They are serious, and they can lead to very, very, maybe even fatal outcomes for a mother. Not so much in Australia these days, but worldwide. This is why women don't survive pregnancy in certain countries. So I would lump those in the category of serious conditions, strong family history. If that is there, then I would test. I wouldn't question it. I would definitely test, because knowing if the woman in front of you who's thinking about having a baby is homozygous or heterozygous, would that make you treat harder? Maybe. I think it would definitely, if she was homozygous for some of these SNPs, if it was CBS and MTHFR, for example, you know that you're going to have to be a little bit more robust with your treatment. If she is, say, heterozygous for CBS and no other polymorphisms related to these methylation pathways are present, you can think to yourself, well, there's a tendency, but she's not in the category of, say, other women who are compound homozygous or compound heterozygous for many different SNPs. So it is a spectrum. It's like most conditions. It's not black or white, but testing is available. It is cheaper. It is available to naturopaths. A lot of testing is so unavailable to us because we have to refer back to a GP or refer to a specialist and write a letter in order to get access to these tests. I think it's a matter of being able to really talk about the validity of the results with your patient and have that really good understanding of saying, look, just because the genes are here doesn't necessarily mean that there's going to be a problem. And we have the tools in naturopathic medicine to, as best as we possibly can, not necessarily override these SNPs, but support your biochemistry and allow your enzymes and your genes and your biochemical pathways to do the right thing at the right time. And there's a lot of counselling that comes into this. You know, there's a whole area of medical science that was born out of all the information in the Human Genome Project. It's called genetic counselling, and it's a whole area of specialisation because it really dredges up a lot of information that we may not know a lot of what to do with it in a clinical situation. And on the other side of the coin, it creates decisions for a couple as to what treatment course they're going to take. And I think we're quite lucky in naturopathic medicine. Some of these SNPs that we investigate in clinic, they don't necessarily have to make huge decisions based on the information that's coming back. They just need to decide whether they're going to adopt the diet that you suggest, and they just need to decide if they're going to adopt the supplement regime that you suggest. They're not being asked to, say, remove ovaries or remove breasts or something, really, really
devastating,
significant
yeah.
that the medical world has to deal with. So I guess, yes, if there's a strong clinical history, I would not be backward in testing. I would go for it.
It's one of those, like I'm still sort of waxing and waning as to if we don't know, and if the SNP doesn't necessarily mean that condition will present. Mm-hmm. In the face of having a strong family history, then again, what's the point of testing? Because you'd still be treating even in the absence of testing. And conversely, if you don't know that somebody's homozygous because they've got a heterozygous SNP from the mother, but you don't know what they're going to get from the father, then conversely, you don't know the seriousness of the potential condition. It's like, yes, which way do we go here?
Yeah, of course. And I think the thing to look at is, well, there's a whole layer on top of our genetics known as epigenetics. And the other thing that might make you lean towards testing would be, what are the epigenetic factors that could be actually bringing these particular SNPs, if they're there, causing them to be more problematic. So if you've got that typical situation of long-standing poor dietary history, long-standing toxicity issues, environmental toxin exposure, a history of smoking in particular, which is a huge drain on all our methylation pathways, ethanol abuse, a huge amount of stress, a microbiome that has basically been deconstructed over decades because of all of those poor dietary habits, environmental, lifestyle factors. If that's all there, and there is also a strong family history, again, testing testing probably becomes important there. Having said all that, you do have this. I need to have this tattooed some. If I ever get a tattoo, it's going to say "Treat what's in front of you."
Yeah.
There's 2 tattoos I'm going to get, and they're both about naturopathy, and that's one of them. It's "Treat what's in front of you."
Yeah.
So if you can see that there's this couple or this woman is thinking about having a child, having a baby, and you've got that diabolical backstory in her family history and a diabolical history of poor diet, poor health, etc., it probably won't change how you're going to treat her.
No, that's right, yeah.
I think possibly it might come into play where you have to use your judgment and possibly the art of naturopathy in, is this going to help with her compliance? Would having a test actually help with patient compliance? Because in many areas, not just in what we're talking about today, but in many areas of naturopathy and medicine, sometimes a test and having that in front of them makes them follow your advice.
In your experience, do you find that testing can either cause guilt, if you like, oh my God, I'm going to hand over this gene to my baby, or do you find, as you've just mentioned, that it can actually help the person to say, right, I now know that there's something that I need to consider managing, and I now have the evidence to say it could potentially be that way. Not that it is, but that it could potentially be that way. In your experience, how do you find patients feel about testing? How do they respond?
Look, there's always apprehension with genetic testing. It's very different, isn't it, to just going and having a full blood count and being checked for anemia, because it's your genes. And a lot of people have that, in the general population, when you talk about genetics, it's something you can't change. It's something you've inherited. It's something that you've got your quota of genes. You've got the information there in your genes, and once whatever is there is present, there's nothing you can do about it. So people are always nervous, I think, is the way that I've found, is that if you say, look, maybe we need to test for this to see how you're going to, say, cope with a particular dietary change or whatever. So there is apprehension, no matter their background. I see a lot of women from scientific backgrounds in my clinic, and they're apprehensive. Right. Even people that work in genetics, they're apprehensive. So there's a lot of, like I said, genetic counselling that comes into this, and you do have to explain things like, well, just because it's there doesn't necessarily mean a clinical outcome is going to be a problem, okay? We talk about epigenetics a lot. I talk about epigenetics a lot with clients, and I give them information to read about epigenetics. I talk about that this is the layer above our genetics that we still know virtually nothing about. So it's all about things like diet, lifestyle, stress, sleep, whether or not we smoke, whether or not we consume a lot of alcohol, drugs, etc. So there is a lot of talking and a lot of counselling that comes into this. And usually if they're worried about passing something on, we also talk about the fact that whatever it's likely to be doing in you, it is also likely to be doing in your offspring. But at the same time, we can talk about the right type of diet, the right type of lifestyle that's going to nullify any potential risks associated with an SNP. And I also talk about the fact that these things have probably been in the human population for goodness knows how long, and it's only now that because we know about it and we can test it that we're even having this conversation. But at the same time, knowledge is power, and if we know that it's there, the way that we address it is simple. There are simple things that we know that can address these issues. A lot of people think that they're mutants, and I have to basically describe what a mutant is and that they're not one of them as well.
You know, again, I'm vacillating because you have on the one hand this, as you say, knowledge is power, and yet you have on the other hand this fear, given even in, you know, people that are au fait with genetics, because they have this preconception of what genetic testing tells you about with regards to, you know, serious conditions like cystic fibrosis, Huntington's chorea, that sort of thing. Now we're getting into the thing of potential, not necessarily actual. And it's sort of, 1 minute I will be saying, yes, test. And the next minute I will go, really? So let's take it this way. If I could say, okay, your total patient population, how many genetic tests have you done, percentage-wise?
Oh, look, I would say it's probably less than 20%.
And how many has it changed the treatment of?
Gosh, probably even less than that, maybe 10%.
10%.
Of the people I see.
Yeah.
And where it has changed things has more been around diet, more so than anything else, more so than heavy dose supplementation in that super physiological level, which I don't really go to anyway in clinic, but really it's more been around dietary changes and permanent for life dietary changes. Okay,
so here's my take on it then. If diet is always the foundation of naturopathic natural treatment and supplements are used judiciously, then if you're using this sort of thing to solidify or cement the importance of a good, well-balanced, vegetable-based diet, then I can only see this as being good for those people that you've done testing for.
And if you talk to any practitioner that does or has used genetic testing in their clinic, you almost have to, you're never going to use it with all of them. You base it always on the family history, the clinical history, but also things like physical examination, their dietary history, the types of medications they're currently on or that they've been on long-term in their past. any questions that come up for you related to their nutrient assimilation. Only then, when you've done all of that background, do you then even think about ordering some of these tests.
Yeah.
And in many cases in clinic, I would actually order routine pathology as surrogate testing and surrogate markers before I ever thought about doing genetic testing.
What do you find the concordance is when you're looking at SNPs with regards to your functional pathology test? Let's say the hydroxy, methoxy estrogens, the 2 to 16, the 4 series.
Look, I think usually if you're seeing abnormalities in more routine or more orthodox testing or even functional pathology testing, if you're seeing imbalances there or abnormalities there, then most of the time if you went looking, you would find a SNP. I don't know I couldn't say what the concordance is with. Most of them are homozygous or most of them are heterozygous or they're compound heterozygous for many SNPs. So I couldn't even think about what the percentage would be there. But if you're seeing abnormalities on routine and functional pathology, if you went looking, you'd probably find a SNP. That's not to say that you would go looking for a SNP if you had abnormalities here. But particularly with routine pathology, as most of the listeners would know, usually if something's showing up on a routine pathology test like bloods or homocysteine being abnormal or even methoxyestrogen, something like that, usually there's a fairly decent amount of pathology underlying that because the body does really like to keep things healthy and keep things balanced. So if something's showing up in, say, blood or serum or even urine, then usually there's a problem. Homeostasis has gone awry somewhere. So I actually find those routine pathologies a great tool to just start with. To start there, just do some serum B12, red cell folate, red cell zinc, a full blood count, looking for things like macrocytosis to give you an idea whether or not B12 and folate is deficient, just doing homocysteine levels on blood, just starting with something like that. And again, doing it based on clinical history. These are not just screening tests that you do with every patient that comes in. It's because you suspect that methylation pathways, COMT, CBS might be implicated here. So, these are surrogate tests, even B6. I didn't even mention B6. But you can do
the much maligned B6.
Yeah, exactly. So you can do these tests in routine pathology, and that can sometimes add weight to your decision with your patient to actually do further, say, genetic testing. I find the biggest problem in clinic is when people come to you with their genetic testing.
Oh, okay.
And they've ordered it over the internet or somewhere.
Yep.
They've had it done, and then they come to you with a paper or a result sheet saying, I've got this. What do we do about it? And then you're sort of working backwards. You then have to do everything that you might not have ever needed to do with them had you seen them at the beginning of the journey and didn't have any genetic testing to go on.
Yeah.
So there's a lot of genetic counselling that comes into that. There's a lot of backtracking that comes into it. You might then have to talk about routine pathology as a way of saying, well, okay, you've got these SNPs, but let's actually see if they're causing a problem for you. If you're concerned, we can do the routine pathology and see if it's actually manifesting in a physiological sense, and we can pick up deficiencies in your bloods or your urine. But at the same time, the treatment will still be the same. It'll still be dietary intervention and lifestyle intervention and looking for any kind of epigenetic factors that might tip the balance in favour of the abnormal effects of the SNP.
You were talking before about epigenetics. I guess we've got to go there with regards to diet, nutrition, lifestyle. I remember Mike Ash saying, if nutrition is really where the pedal hits the metal, should we just be talking about nutrition anyway with regards to epigenetics, because we're doing it anyway? We should be advocating a good, healthy diet and lifestyle anyway. So what, therefore, is the facility of testing? However, there's this backflip about it gives you knowledge to say, I should be leaning this way with regards to my diet. So with regards to epigenetics, how do you decide what focus to take with regards to diet?
I think it depends. There's a little bit of how someone practices that comes into this a lot, I think. For me and how I practice, diet is everything, no matter what treatment you need to do for someone or what conditions need to be corrected. It's always diet first. Now, my belief is that because we've gotten so far away from our evolutionary diet, and this is just all my theories, because we've gotten so far away from our evolutionary diet, these SNPs have more of a chance to cause problems. Now, the problem I'm finding is that when people present and then you start talking about getting back to, say, a more evolutionary-based diet, there are compliance issues. There are issues around time and how long it takes to actually prepare meals that allow you to follow an evolutionary diet. There are social issues that surround why people won't follow an evolutionary diet. But I truly believe, and there's no science to back this up, but I truly believe that if we were supplying our body with, great quality food, the correct amount of protein, fat, good carbohydrates, if we didn't have all the issues that we have with the food supply that we now have, I really don't think that these SNPs would be really manifesting clinically as much as they are now.
Oh, for sure.
But
Absolutely.
So what we have is a bit of a conundrum because there's all these different reasons why people can't or won't follow an evolutionary diet. And so that's why we're having to talk about things like, say, epigenetics and stress comes into this as well. And that's why we're sort of talking about the idea of maybe having to go and modify enzyme activity with high-dose supplementation. So for me, I think the epigenetic storm that can happen from this is never going to be as problematic if the foundation underneath, which is food, it's not, for me, herbs are medicines. They're not something that provides a foundation to our health. It's not, nutrition provides the substances that actually build our body, that builds our genes, that builds our enzymes, that builds our neurotransmitters. And if that foundation is correct, then the epigenetic insults usually wouldn't be as severe when they come. And the other thing is modifying those epigenetic insults as much as we possibly can. Nutrition and sound nutrition is the buffer that allows us to deal with those epigenetic insults that are going to come. There's always going to be times of stress. There's always going to be times of imbalance. But if that foundation is solid, we tend to weather those storms a lot more easily.
You mentioned getting away from our ancestral diet. We've also gotten away from our ancestral lifestyle as well, though. The production of adrenaline in ancient times would have been rather acute to save your body from death, to get away from that lion. Nowadays, it's to get away from rent or marital stress or work stress.
It's all of those factors, food and lifestyle and the types of stresses, the way we sleep, everything is involved.
Yeah. So then can I ask you, when you're talking to patients about dietary intervention, do you ask them how much they're willing to give, how much they're willing to change?
Yes.
How do you approach that? And I've got to ask, how do you get them to commit? And how do you find that, how successful do you find that?
Usually a little bit of, I guess, getting away from the whole, this is all my knowledge in the sciencey part of it, and just talking to them as one human to another. And it's almost like you just want to take the practitioner hat off for a little while and say, look, these are not easy things to do. people being asked to change their diet, possibly for the rest of their life. These are not easy things to do. So you have to talk about things like willpower and commitment. And I actually read a lot. I'm reading more to help me be a better practitioner. I'm reading more about how to talk to people as opposed to all my biochemistry stuff, because I'm finding that makes so much more of a difference. So I recently read about this idea in psychology that willpower is absolutely bollocks. It's not going to get people anywhere, and that the way that you can get people to adopt any kind of change, whether it's more exercise, changing a diet, getting to bed earlier, whatever it is, giving up alcohol, whatever it is, it's not about willpower. It's actually about working out your why and saying, well, why do you want to do this? If you can, and make that decision, make it public, have people that make you accountable for this. Don't just decide in your head, oh, I'm going to have really hard willpower and I'm not going to, all this stuff's going to be in front of me, but I'm going to try really hard to not do this. I talk to people about their environment, and I've started doing this a lot more recently, where I talk about the fact that you can create your own environment. Yeah. If you want to change and that's your why is because you want to be healthier and be around your kids and be alive for as long as possible or whatever it is, you have to change your environment, because willpower is really hard to stick to long term. It might work for a few months, but it may not work forever. It's about changing the environment and getting people on board with you, and not just sort of making that decision in your head, but telling people around you, telling your family, telling your friends. And often you find that if there's one person in the family that you're treating, I try and focus on what are the rest of the family doing, because sometimes by default, if the family comes on board with a change in diet, the whole family gets better by default, which a lot of practitioners know anyway.
Yeah.
So
I'm so glad you said that, though, that sentence about find your why. That's indeed how I gave up smoking. I remember exactly the time, because I found my why.
Yes. I can't even think of who the author is at the moment, but because I'm reading a whole bunch of stuff and I don't know who to, but it's Find Your Why.
Find Your Why.
And so we sit in clinic and I say, if you don't know what your why is at the moment, that's okay. You might have an epiphany on the train on the way home, or in 2 weeks' time you might be in a situation you think, well, this is my why. Yeah. And next time we speak, let's talk about it, or send me an email and tell me you found your why, and that's fantastic. But at least have a think about it, because if we don't really know our why for doing something, we don't change the environment to set ourselves up for that success. And, there's a lot of human psychology that goes into, as we all know, that's the art of naturopathy and nutrition, which we couldn't, that's a lifelong skill. That's not something that we get great at just because we've graduated or been in practice for 20 years. You're constantly working on it.
But it's such beautiful art that you practice. Well done. I've got to ask regarding diet, and if we're talking again about epigenetics, and we're talking about modifying the activity of SNPs or the downstream issues of the SNPs. Yes. How hard do you go with diet? How long do you treat? Do you ever do a short-term intervention and then modulate the diet longer term?
I guess the short answer to that is say it's a scenario where you don't have any genetic testing in front of you and you're just going based on a clinical hunch, or not even a hunch, but clinical evidence of the person in front of you. Always diet, and always hard in those first few months, and then modified as you, I guess, get feedback from the person as to, you have to think about why they're coming in. So they might be coming in for a mental health issue, they might be coming in for fertility issues, they might be coming in for any number of reasons related to COMT, CBS, methylation. And if you don't have that genetic information in front of you, you start with diet. I wouldn't even start with supplements at this stage. You just start with diet and you get feedback. You start to get feedback on the things that you're measuring. So you might be measuring anxiety, or you might be measuring something like, if it's a fertility thing, you might be measuring menses and regularity. You've always got those things that you're trying to modify, and you're measuring those outcomes. So definitely always diet. You would go harder, I think, in terms of supplementation if you had something like seriousness of condition, so something like history of embolus formation, really if it was severe anxiety, maybe severe ADHD, severe maybe depression, if you're suspecting COMT and these sorts of things, and you've got some testing in front of you, that's when you might start thinking about supplementation. For me personally, I don't really go into that super physiological dosing range that's very, I won't say popular, but it's out there. There's been some great research on modifying enzyme activity, either upregulating or downregulating with this super physiological dosing regimes of supplements. But for me, I think and I've seen it in clinic, you can still get really good results. If you're working on the diet and they're compliant, you don't need to go down that path of really hitting it hard with supplementation. And the other thing with that really heavy high-dose supplementation is that it often can make people worse, and I've seen it. Yeah. Particularly if you're talking about COMT and anything to do with mental health issues. Often their anxiety worsens, their depression worsens, their panic disorder worsens, and then you've actually got to pull back. The other thing that happens is, particularly with something like COMT and it's a mental health issue, is you've also got an inflammatory issue on top of that, say joints or arthritis or something. If you start going in with, say, something like, heavy SAMI or something, it flares up. And I'm a fan of the gently, softly approach. That's just me and how I've always practiced. I'm not particularly gung-ho with high-dose supplementation. So you have to get a lot of feedback. You need to be in close, I guess, communication with someone and asking them, sometimes even on a daily basis, just checking in with a quick phone call, how are you feeling today? Or call me at this time, every couple of days, and just let me know how you're going. Because that's the feedback that we need. We can't just say to someone, see you in a month or see you in 2 months, come back. If you are hitting hard because you've got genetic evidence or really strong clinical manifestation of serious problems, you do need to be in that really, really close contact. The other time that I would suggest maybe hitting something hard with, say, diets or supplements is if other treatments have failed. If they've tried diet alone and they've been really compliant and they've tried it for a while and they've got at least, say, 6 to 12 months of dietary change, and you think that their compliance has been good and they're telling you it's been good and they've tried other things and it's not working, diet alone, then you might think, okay, well, now we need to talk about supplementation and we need to go a little bit harder here. But in terms of high-dose supplementation long-term, I really don't think that there's any real evidence for that. Different clinicians, there's clinicians all over the world and researchers working on this. And a lot of this is still weak genetic data, to be honest. It's not strong genetic data. We don't know the long-term safety of these super physiological doses if people were to follow it for 5 years, for example. We just don't know. Yet we're being expected to adopt it in our clinic. That's the hard thing. It's almost like the information is coming out so fast, and every 6 months there's new studies and new seminars and new protocols. but we don't know.
And so I do totally agree with you that we should always be checking in with our patients and finding out if, what little they can be on, if at all. I have to ask, just as a last question, we spoke about ethical issues just previously, and I have this question. If somebody does present to you with their already done test, is that considered a pre-existing condition because they now know what their genetics are, even given that the SNP may or may not have an effect? Is that considered legally a pre-existing condition?
That's a very good question that I don't know the answer to. I think because it's a SNP, I doubt that it would be considered a pre-existing condition. To my scientist mind, I would be looking for, if I had to make an evaluation on this as a medical ethicist, I'd be thinking, well, is it homozygous? What's the evidence? If there's not a lot of scientific evidence out there, it weakens the argument for pre-existing condition.
Yeah.
And the other thing is we need to be looking at who did the testing and the methodology that they used. So who was the lab that offered the test? Was it a bit of a garden variety or even a sort of a backyard setup where the testing methodology wasn't legitimate? Or is this a legitimate laboratory offering legitimate genetic testing? Because they're not all the same.
No, that's very interesting. There's a can of worms that I didn't know about. I thought it was all, here's your chip, and it's sort of basically that's what you get your testing done on. So it's not that. There is variance.
No, there are definitely some services out there that sort of they're laboratories, but there's all different methodologies for this, and some of the methodologies have been superseded by more accurate methodologies, and the older ones might still be being used. Right.
So false positives, false negatives?
Both.
Wow.
To be honest, but more often than not, it might be something like a false positive.
Right.
More often than not, and we're also talking about things like within laboratories, as you would know, quality control is a huge issue. So laboratories that are part of a formalised quality control program are expected to have their own internal quality control that they then report to the overseeing quality control body so that they are monitored. So there are laboratories out there that might be offering legitimate methodologies, but the accuracy of their results may decline over time because they don't have that strong quality control process internally. And, it costs money to be part of such a program, but obviously you should be part of that sort of program as a lab because we're talking about people's health data
that's right.
here. So,
that's exactly right. Annalies Corse, thank you so much again. I mean, this is, you talk about a tip of the iceberg with this, and we've just covered 2 genes, and even then we've just really tapped it on the outside of the iceberg.
Scratch the
surface, not even the tip of the iceberg. We've only scratched the surface of the tip of the iceberg. But I do thank you for taking us through, the relevance, if you like, of testing. And I do really admire you for teaching us the real ethical stance that one should take as a practitioner when you're looking at, whether you're going to test or not, and indeed how you're going to treat people. So thank you very much for taking us through this today.
Thank you so much. Thanks for the opportunity.
This is FX Medicine. I'm Andrew Whitfield Cook. If you're loving our FX Medicine podcasts, please don't forget to share us with your colleagues, family and friends.